look like if I went toward the tip? So if you did a section through the apical meristem. Well, those are going to be the ground meristem, procambium, protoderm. Well, what happens if we started to do a section in which you started the secondary growth? So now it's going over here to my drawing that we just began here. And so it's showing you the vascular cambium is now in a complete circle. What happens now once you start to get that new cell division? This is how you get this complete circle of secondary xylem and phloem, and this is what it would look like after the first year. And you've also had the periderm formed, which we haven't really covered yet. And then over here, it's a diagram to show this more down the sides, what's happening. But between these drawings, some people like one versus the other. I give you both to hopefully help you visually see what you have looked at on these cross sections. And the only way to make sense of this is you need to look at the visuals, read the textbook, work through it, and then lecture next week. I'll help you through it some more. Repetition is good. And then the less you're memorizing, the better. Okay, anything before we look at this? Okay, this is a tree that would be roughly how many years old? 2. I'm going to start at the pith. Here's the pith. That's this. And from here to there, this bracket is one growth ring. It's one growth ring or one annual ring of secondary xylem. And here's the second one. And then what's this line of tissue here at the arrow? There and there. Vascular cambium. And now if I come down here, what is everything to the outside of the vascular cambium? Not periderm, because you forgot there's phloem in there. Bark.
Oh.
Sounds like a dog. Okay. Okay, so this is bark. Now, some people think bark is the same thing as periderm. Well, notice, look, here's periderm. Well, this is secondary phloem, so don't leave out the secondary phloem. So if I come back up here, here's secondary phloem. And this is the remains of the cortex. And now here's the periderm. Now, you were asking me, is how long is the cortex going to be there? Well, here's 2 years old. The cortex is still apparent. That's the same cortex as it was right here on this drawing where it was put. Here's the pith, and here's the cortex. And the cortex doesn't just suddenly disintegrate. It's still there after a couple of years. Well, I put this picture here because in the lab it said, Lincoln has people ask me, I said, "What? There's crystals in the parenchyma." Yeah, especially after you get a tree a few years old, it is, you'll see lots of crystals. Now, the crystals can be a lot of chemicals. So notice that these crystals aren't like the little toothpicks. Different chemicals crystallize differently. So don't be surprised is when you're looking at parenchyma within the tree, the rays, you can see all kind of shapes of chemicals in there. And that's where they've concentrated so much that they have crystallized or precipitated. Okay, this is the same thing we had before, but now I've increased magnification. It's looking toward the outside of the tree. And then what would be this single cell layer there? These two arrows are pointing at. That would be a xylem ray. And then that circle there, it's really pointing to the center of what's likely a vessel. So most of this is likely vessels, but at this point I'm not asking you to distinguish. It's certainly a conducting cell, vessel, or trachea. And then, can you tell at this magnification what you've seen before, all this pinkish stuff? Not parenchyma fibers. Is this, are there fibers, parenchyma tissue? It's, or more specifically, it's phloem fibers. Because this is in the phloem, so we'll call them phloem fibers. Now, if you look, here's phloem fibers, phloem fibers, phloem fibers. Can you see there in layers? What's between the layers that this arrow is pointing at?
My battery's dying.
Companion cells and sieve cells. Yes. And what kind of tissue is this? Parenchyma, and it's an array too. Notice the rays in the phloem get a lot bigger for reasons I'll let you think about, and we'll cover next week, but I don't want to clutter your mind at this point. But I want to emphasize that this is the first time that you should easily see all four cell types in phloem, because we've said since chapter 3 that phloem can have parenchyma, phloem can have fibers, phloem can have sieve cells or sieve tube elements, and companion cells for the albumin cells. And here all four are. And they're really easy to see in this. And what makes it easy is the parenchyma usually occurs in big clumps in array. And the fibers alternate with the conducting cells and the support cells. And then they're really very different. And once you leave the phloem, so if you keep coming out the phloem, you can see it abruptly stops. And now I've left secondary phloem and I'm in the cortex.
So if that was on a test, a picture, and you're pointing at the fibers, what do you want for an answer? Because they are.
Well, if I wanted you to say fibers, I would hope I wouldn't ask it here. What I would do is I would go to this and say, I'm going to ask you a question about something out here. Let's magnify it some more. Okay, here it is on 100x. Okay, now let's go to 400x. I'm not going to show something to you and just let you guess, but I figured you could figure it out here. But on the test, I would magnify it enough that you could see it. And then if I wanted you to say fibers, and I had this magnified, which I don't think I have right now, if I wanted you to say fibers, I'd say, name this cell type that's found here, not the tissue type.
So would that be
Fibers.
the answer is fibers?
If I said cell type versus tissue type.
Okay.
If I wanted you to say sclerenchyma, I'd say, tell me what simple tissue this is. Not, the reason I'm saying simple tissue, because if I just say, name this tissue, you could say, correctly, sclerenchyma, or you could say phloem. Because phloem's a complex tissue. So, if I say simple tissue, it can only be sclerenchyma. If I said, well, what's the complex tissue, like all of this, then it would be phloem. Or if you said secondary phloem, so much the better. That would be more accurate. I see. Now I don't have a magnified view of the fibers because we covered that before. Here, now we already covered this. Is there any problems here? Vascular cambium, secondary phloem, secondary xylem, periderm. Oh, here's one thing we didn't go over before. Notice within the secondary xylem, can you see that this portion in here is a little bit lighter colored, and this is getting darker, and you can actually see that quite well in the samples over here. What's the lighter portion which represents the younger secondary xylem? Sapwood, and then this is heartwood. And the reason for this, and I'll just briefly mention it, this is the lecture material. Heartwood is older and it's darker usually because all 100% of the cells are dead. There's no conduction whatsoever, no water conduction, and the plant secretes chemicals that darken the wood. It's called heartwood, and that makes better lumber. Cheap lumber is made from sapwood, and it's not as durable and it rots more easily, and it's the wood that in a tree that you have still some living cells, but that's where the active conduction occurs. And especially on the wood samples I have over there, you can see it, that there's a distinct two bands or two colors in the xylem, the sapwood and the heartwood. Now, since I have it here, I wanted to ask is, can you see, I know those of you toward the front can you see the little dark circle up there? Mm-hmm. Any idea what that is? It's springwood. It's the same as, where's my best picture? Oh, right here. See those big circles there? That's springwood. And summer wood, the growth slows down. It's getting hot in summer, not as much water, growth gets slower. But some of these vessels get so big in springwood you can see them without magnification. And here